In this week's news, we'll be reporting the top stories presented in Vienna at the 2015 European Cancer Congress. All this and more starts right now on OncLive News Network. Hello and welcome. I'm Laura Jones. Today, we'll start with two big trials in kidney cancer. First off, the excitement continues over immunotherapy in metastatic cancers. In results of the CheckMate 025 trial, nivolumab reduced the risk of death by 27% when compared with everolimus in patients with advanced renal cell carcinoma. After a minimum 14 months of follow-up, overall survival was 25 months for nivolumab versus 19.6 months for everolimus. Progression-free survival was also better for nivolumab, as was the rate of grade 3 or 4 toxicity, which was 19% for nivolumab versus 37% for everolimus. Study author Padmani Sharma said the results are significant and clinically meaningful to patients and healthcare professionals alike. They're likely to change the treatment of patients with advanced kidney cancer whose disease has progressed on prior treatment. Also in advanced kidney cancer, the multi-kinase inhibitor cabozantinib showed favorable progression-free survival when compared with everolimus at 7.4 versus 3.8 months, the hazard ratio translating to a 42% decrease in the risk of progression or death. The patients in the study had progressive disease after anti-VEGF TKI therapy, with almost a third having received 2 prior TKIs. Lead investigator Dr. Tony Schwery from the Dana-Farber Cancer Center had this to say.
Cabozantinib is a dual VEGF inhibitor, also MET inhibitor and XL inhibitor, and these two important targets have been involved in resistance to VEGF inhibitors and to progression in preclinical models on VEGF inhibitors, as well as poor prognosis. So cabozantinib target multiple kinases, including the VEGF receptor as well as MET and XL. So it's an unmet need. VEGF resistance is an unmet need. So the trial asked the question, is progression-free survival, the primary endpoint on the METEOR trial, superior with cabozantinib over everolimus? And now at the European Cancer Congress, we have the result presented yesterday showing that cabozantinib is associated with a 42% reduction in the risk of death or progression compared to everolimus. This corresponds to a highly statistically p-value less than 0.001. So the trial met its primary endpoint.
The data will be under review by FDA as part of the breakthrough designation already awarded to cabozantinib for use in this setting. Next, everolimus will likely become a new standard for the treatment of advanced non-functional lung and gastrointestinal neuroendocrine tumors, or NETs. In a phase 3 study, median progression-free survival was 11 months on everolimus compared with 3.9 months for placebo, as assessed by central review. This was then confirmed by investigator assessment at 14 months versus 5.5 months with placebo. We found a statistically significant 52% reduction in the risk of progression or death in favor of everolimus, and also a clinically meaningful 2.8-fold improvement in PFS, said lead investigator James Yao from MD Anderson Cancer Center in Houston. Although we knew from previous studies that everolimus could delay the growth of pancreatic NETs, this is the first time we have been able to conclusively show that it is effective in other NET sites. We hope our results will provide a new treatment option for lung and gastrointestinal NETs. Also, for patients with advanced midgut neuroendocrine tumors, the novel peptide receptor radionuclide therapy Lutathera significantly increased progression-free survival over octreotide LAR in the NETTER-1 trial. Lutathera works by attaching to somatostatin receptors to release a cytotoxic radiotherapy directly to the tumor. Dr. Philippe Rezneski from the University of Paris discussed the results of his study.
So the main finding in this group of 230 patients half being treated with lutetium, the other half in the control arm receiving octreotide LAR 60 milligrams every 28 days, was that there was a very, very significant benefit in progression-free survival in patients treated with lutate. Indeed, PFS was 8.4 months in the octreotide 60 milligram group and was not reached in the lutate group with a hazard ratio of 0.21.
Overall, the agent was found to be safe with tolerable toxicity. Next, let's move on to lung cancer, where there now appears to be an effective targeted therapy for patients with relapsed and refractory small cell lung cancer. The antibody drug conjugate rovapatuzumab tesirine, or Rova-T, showed activity across a range of patients, representing the first demonstration of efficacy for a targeted therapy in this setting. The new drug also has a predictive biomarker called delta-like protein 3, or DLL3, which is expressed in about 70% of small cell tumors. In the phase 1 study of 73 patients, Rova-T was associated with an objective response rate of 64% in patients whose tumors were sensitive to first-line combination chemotherapy and were positive for DLL3 gene expression. In the third-line setting, where no standard treatment is currently available, the overall response rate for DLL3 positive patients was 45%. It was 20% for all patients regardless of DLL3 expression. As a second-line therapy, Rova-T had an overall response rate of 44% in DLL3 positive patients, 24% for all patients. Side effects are similar to those of chemotherapy. In addition, pleural effusion and photosensitivity were notable. Also in lung cancer, treatment with nivolumab reduced the risk of death by 28% compared with docetaxel in patients with previously treated non-squamous non-small cell lung cancer who were enrolled in the phase 3 CheckMate 057 study. According to updated results, after a minimum follow-up of 17 months, the overall survival rate with nivolumab was 39% compared with 23% for docetaxel. Median survival was 12.2 versus 9.4 months. 1-year progression-free survivals were 18.5% versus 8%, and the objective response rates were 19% versus 12%. Adverse events were significantly less frequent with nivolumab than docetaxel, reported in 69% of nivolumab and 88% of docetaxel-treated patients. Grade 3 and 4 toxicities occurred in only 10% of patients treated with nivolumab, compared with 54% for docetaxel. Under the expedited review process, the FDA's decision deadline for nivolumab in previously treated non-squamous non-small cell lung cancer is January 2, 2016. Another checkpoint inhibitor, this time a PD-L1 inhibitor called atezolizumab, formerly MPDL3280A, demonstrated significant clinical efficacy as monotherapy in patients with advanced non-small cell lung cancer. In the phase 2 BIRCH trial, more than 61% of responses were ongoing after a minimum follow-up of 6 months. Tumor shrinkage was observed in up to 27% of patients whose disease progressed after prior treatment and who expressed PD-L1 at the highest levels. Higher PD-L1 expression correlates with higher responses and may allow identification of patients most likely to benefit from atezolizumab, said lead investigator Benjamin Bess. Previously, the phase 2 Poplar trial showed a median survival of 15.5 months with atezolizumab in high PD-L1 expressors compared with 11 months with docetaxel. Genentech is currently in discussions with the FDA on the submission of data from the Poplar and BIRCH. In another study looking at atezolizumab, this time in metastatic bladder cancer, the PD-L1 inhibitor demonstrated significant clinical benefit in heavily pretreated patients. Lead investigator Jonathan Rosenberg, a medical oncologist at Memorial Sloan Kettering Cancer Center in New York, commented that the status of available treatment in metastatic urothelial cancer is dismal, quite frankly. No single agent has improved median overall survival. Vinfluinine is the only approved agent in the European Union and demonstrated an overall response rate of just 8.6% versus 0% for best supportive care. A phase 2 trial of atezolizumab enrolled all comers and showed that in the first-line setting, for those whose tumors were PD-L1 positive, an overall response rate of 26%. In the second and third-line setting, the overall response rates with atezolizumab were 39% and 20%. The median duration response has not been reached. Median PFS at 24 weeks was 2.1 months across all groups. In our last story from this year's European Cancer Congress, the combination of dabrafenib plus trametinib improved overall survival by 7.6 months compared with single agents vemurafenib in patients with unresectable or metastatic BRAF V600E or K-mutant melanoma, according to the 2-year analysis of the Phase 3 COMBI-V study. The median survival with dabrafenib and trametinib was 25.6 months versus 18 months with vemurafenib alone. Estimated 2-year overall survival was 51% with the combination versus 38% for vemurafenib monotherapy. A similar story came out of the recent COMBI-D trial, in which the combination of BRAF and MEK inhibition was associated with a 25-month survival versus 19 months with dabrafenib alone. And that's going to do it for today. Thanks so much for watching OncLive News Network. I'm Laura Jones. We'll see you next time.